Hello, everybody. Welcome to our meeting, to our webinar. This is Andreas Papas, and I am a veterinary student from Greece, and I am an IVSA Thessaly member and the chair of Standing Committee on One Health. Here today, you are to hear the webinar about food safety and COVID-19. So, our lecturer for today is the director, Dr. Francisco Diez-Gonzalez. Is the director of the Center of Food Safety and a professor in the Department of Food Safety and Technology at the University of Georgia since 2016. What happened actually was that he made a webinar like this one, a similar one, 2 weeks ago. And I took the idea to start a small series of webinars. We will have one every Saturday. We will do as many as we can. And as I took the idea from him, I suggested him to be the first lecturer for today. The next person that you will meet today is Nabila, and she is the secretary of Standing Committee on One Health, and she will coordinate the rest of the webinar. She will help you also with technical problems if you have any. And this is our webinar. Before we start, I want to tell you that you are welcome to participate in any campaigns that we do the rest of the term. And you can find all information about them in our social media. I want also to remind you about our event in Turkey, in IVSA SCOH will host that. And this event actually was to be today, but it was canceled because of the virus. So we decided to postpone it. You will take further information about it through our social media. We have separate social media about the event, so you can follow us there too. So now Nabila will unmute our lecturer. And thank you very much, Dr. Francisco, for coming and helping us with this event.
Well, good afternoon, or good evening, wherever you are. I understand that you're an international group from what Andreas told me, and thank you for the invitation. And I'm going to try to share with you some insights on the topic of in everyone's minds during this period of time. So let me then. I'll start sharing my screen then. Okay, can everyone see this first slide? Okay. Okay, so I think this is as Andrea said, this is just I'm in some way replicating what we did a couple of weeks ago with a group of students in Georgia, and there was interest on coronavirus and food. So we had this which is not really a seminar in the strict sense, because the initial part is just to give you an introduction of who I am and what research institution I am representing. But more than anything, it's going to be a session of that I hope that you have the opportunity to address some of your questions or concerns. So that's mostly what I hope we're going to be accomplishing today with this exchange or this webinar. So the first few slides is basically the slides that I share with Andreas. Is basically, it talks about our center, and I guess it's going to lead eventually to our exchange of questions and answers. So so we're here in the middle of this historical pandemic in human history, in which I guess everyone is affected at this point. Pretty much every single life and everyone in the planet has been affected. So we're going to be dealing with this virus for quite a while. Anyway, first of all, as Andrea said, I'm at the University of Georgia. I've been as director of the Center for Food Safety for approximately almost 4 years. And before that, I did most of my academic career at the University of Minnesota in the United States. I'm originally from Mexico. That's where I got my accent from. I've been in the US for for the last 30 years. First, I came to pursue my graduate school, and then I stayed eventually for the rest of my career. So just if as you may know, Georgia is one of the states in the United States. We're in what is referred to southeast region of the country, and we're located in one of the campuses of the University of Georgia, which is Griffin. The main campus of our university is in another city called Athens. If you're from Greece, like Andreas, so you will realize there are actually multiple Athens all over the world. In this country, at least all the neighboring states of Georgia have at least one town called Athens. So those are just a few pictures of our campus just to get to know and a little bit more. And our campus was originally founded as an experiment station. And this is a little bit of history, but we're not going to dwell too much on this information, just to give you an update where we're located. The Center for Food Safety is an academic unit from the University of Georgia that's part of one of the College of Agriculture and Environmental Sciences. And this center has existed for, it's going to be more than 27 years. It was established in 1993, and it was originally intended for not only food safety, but also for quality enhancement. My predecessor in this position was someone that is a food microbiologist with a great reputation in the world. He was very actively involved when E. coli 157 came into the emerge as a foodborne pathogen. And then eventually the center changed or shifted its entire focus on food safety alone. That was 2001. Our center is basically focused on research in foodborne and anything related to foodborne pathogens, either prevention, detection, and control. And we are working very closely with government and industry groups. We do this mostly through basic and applied research, but we do have quite a bit of outreach in linking up different groups, mostly industry groups and government groups. We also are involved in education because we train graduate students in our microbiology laboratories. No, it's this thing is here. Someone Zoom bombing me. That's really odd. I don't know if you're seeing a circle in my slide. But, so the center has several areas of interest, which are on detection methods, genomic epidemiology. We have a great emphasis in particular in Georgia. If you don't know this, but Georgia is probably one of the regions that has the greatest concentration of chickens in the world. If you just rank Georgia as a single, let's say you put it compared with other countries in the planet, Georgia would be ranked number 6 in poultry production. For those of you who are veterinarians, I think this particular fact could be of interest for you. And we have a number of other areas that we're working on: detection methods, antimicrobial interventions, and also we have a recent hire that works on antimicrobial resistance. The role that we play is linking up a good number of different stakeholders, mostly in relatively recognizable brands. There are companies that they have footprint in different parts of the world and the U.S., and also with our government agencies, mostly with the CDC. Our faculty in our center, we are all 100% research. Even we don't teach. We're entirely dedicated our research on research. I'm going to go quickly through this so we can go questions. These are the individuals that make up our faculty. We have a total of 8 faculty in our group, and different people are involved in different areas. Dr. Shanyu Deng and Hengden Bakker, they work on bioinformatics and genomic epidemiology, which has been really crucial during this time. And then we have Govinda Kumar, Fresh Produce, Ines Ortega on parasites, is Matt Kasem, who's working in Campylobacter, poultry, and antimicrobial resistant. And I'm going to talk in a minute a little more on Malachi, who is our virologist. Someone that I think is important for you to know is our center has a lot of reputation thanks to people like Larry Bushaw, who's an investigator that's still active in our center. And because it's someone like in his career had more than 500 publications, peer-reviewed publications under as an author called. We have a number of other faculty affiliated with our center that also that provides a lot of strength to our discipline and brings different also different perspectives and allows us to conduct different kinds of research. So that's something is very beneficial. And here I'm just going to list the names, and you can check it up on the university website. I brag about that UGA, University of Georgia, has one of the largest numbers of food microbiologists with emphasis on foodborne pathogens at least in the US. We're linking up with also other institutions. We have faculty that are part of CDC, USDA, and other universities. We have a large biosafety level 2 laboratory. It's about 40,000 square feet. Was built in '96, and it can house as many as 10 individual research groups. So we have plenty of space to work on. This is just an idea. So the space in our laboratories and some of the equipment we have. And I think that's enough for me to talk about our center. That can give you some perspective of where I'm coming from. Myself, I've done quite a bit of research in different areas related to different foodborne pathogens. In linking up with veterinarians in my early years, I worked with E. coli 157 in cattle. So I collaborated with a number of veterinarians back in the early 2000s, and we did a number of projects related to shedding of E. coli 157 in cattle populations. I think, and what our center has been doing during the pandemic? Well, I've personally been quite busy because I've been linking up the food companies with the CDC, and it's been a very evolving situation with trying to address all the needs of preventing transmission and making sure that companies are continue to operate during this time. I've been linking up with the media too, giving interviews with different news outlets. And despite the fact that coronavirus is not considered to be a foodborne pathogen, so we are starting research on SARS-CoV-2 because we recognize the importance of this organism for the entire planet, and we also have been able to identify opportunities that, based on our communications with the companies that are associated with our center, there is multiple questions that need to be answered, and some of those questions we're going to be discussing in today's webinar. So the person I wanted to introduce is Dr. Malachi, who's a relatively new faculty in our center. She joined in 2019. And traditionally, she's been a food virologist with emphasis on norovirus. And she worked at one point during one of her graduate degrees with the animal coronavirus. But she was not really doing any work with any human coronavirus, but because of this situation, we're moving quickly to make sure that we provide the resources for her to start working on looking at different areas of aspects of SARS-CoV-2, such as survival and the influence of different factors and the influence of food matrices. There are many questions that we often get from our companies affiliated with us. For example, if someone that is sick touches a package of food, well, whether the virus is going to remain on the surface of the food by the time someone else picks it up. So there are tons of questions that we need to address. So I think and let me, one other thing that I'm going to do is here is I'm going to still leave those slides, but I think I'm going to activate my screen so at least you can see me. Because the next phase of this session today is basically try to fill some of your questions. I have a list of bullet points here that we can go through them if you want. But what worked really well when we had the same session in Georgia with the students at the University of Georgia is basically opening up for questions. I guess that's one thing that worked really well. I don't think back then, if Andreas was there, we did not really go through this list of bullets. I think all of this in many ways came up based on your own interests and your own concerns. And I'm sure many of you, it's really amazing that in the time that we're living, everyone is becoming an expert in virology as we speak, because we're eager to learn a lot, as much as we can. And probably I'm not going to be able to give you the answers, and maybe you're already read about something about a particular situation. I am not a virologist, first of all. Instead of my discipline, I consider myself a food bacteriologist. I've worked with Listeria, Salmonella, E. coli 157, Clostridium perfringens, and other organisms. So I just similar to your case, I've been learning a lot about viruses. And the closest that I work in my experience to viruses is I had a project with bacteriophages, but intended for the traditional foodborne pathogen. So anyway, I think I think since I will open up right now for your questions, and I don't know, Nabila or Andres, how you want to handle the questions, whether you're going to handle entirely by written questions or you're going to allow people to open up their microphones and ask the questions themselves.
Okay, so Doc, I think if it's all right with you, could we please go through, I think I'd really appreciate it if we could go through those points very briefly on your slide, and then I'll be more than happy to open the floor to all our participants to ask their questions. And then we do have some questions that have been submitted earlier that we can ask you in the end.
Sounds perfect. Yeah, I'll be happy to go through these bullet points.
Thank
you.
The
first question that you've seen many experts, and there are the WHO, the CDC, and I imagine wherever wherever you are in the world, your government has been clarifying the fact that that the food is not going to be a vehicle for transmission of coronavirus. That if you consume food that is contaminated, that for the most part, that's not going to lead to infection. And this is basically, I guess, an extrapolation based on other respiratory viruses that normally food is not transmitted. The kind of typical symptoms that coronavirus causes, it is still, the evidence so far is still pointing out that it's direct contact, or at least relatively close contact with another individual that is driving most of the transmission in the current pandemic. So that's one thing that we don't have evidence to believe that food is a vehicle for transmission. I mean, if you ingest it, that's the thing that we're going to be make the clarification. Now, at the same time, one of the questions that we're trying to answer from extrapolating from other viruses, and if you want to look at data of that from other viruses, the closest obviously is the previous SARS. That's the closest virus to this coronavirus. because other people, you may look at data in the literature from other coronavirus. The surrogate organisms, or as because we don't have the data yet for this virus. Look at SARS research that was done previously. So, and you're starting to see some publications on this SARS-CoV-2 already. There are different publications, and the most famous one is one that look into survival on cardboard, survival on plastic surfaces, survival on metal, and yeah, they're starting to come up those results. Now the question is, in the time that we're living, is should we use a single study to drive policy, to make recommendations to the public based on what a single study published? I think it's a little premature, but we don't have anything else. I guess the challenge that we're facing is that we need to advance science in a rush, because we don't have evidence to be able to provide answers. So survival is a big question that we're trying to address. The question is, what happens if somebody sneezes in the supermarket? You go an hour later and grab the same surface where the individual that sneezed grabs. Am I going to get it or not? That's a big question. Now, in our field, I guess one of the questions that that is being asked a lot is, well, what is the survival during food processing? Well, for the most part, this seems to be as a coronavirus, it seems to be a virus that's relatively susceptible to heating. So, and some of the earliest papers that come out, yeah, you can kill it relatively easily with heat. If you cook food, that's your best line of protection, cooking food. It survives well at cold temperatures. That's what the earliest research is pointing out. But if you hit at least at the pasteurization level temperature, 65 Celsius or higher, you're gonna inactivate it really quickly. So that's pretty much in terms of food processing. If you cook something, you're going to be very well protected, even disregard whether you're going to be transmitted through either through contact with the food. The other aspect that we're also looking, what are the conditions what conditions lead to the inactivation of the virus, either in the environment, in aerosol, again on surfaces. And for example, there was the first publication that looked at this. They look at pH, for example. And it seems that this, to be an envelope virus, seems to be a little more resistant than others because it doesn't die at pH 3. The data seems to indicate that it survives well at almost any pH from 3 to 10 that the research has tested. So it's yet to be determined whether UV light and in food processing related surfaces, we're asking the question, what kind of sanitizers or what kind of disinfectant we could use. And for the most part, the early research seems to indicate that most of the traditional chlorine-based or oxidant-based, hydrogen peroxide, even quaternary ammonium, they seem to be effective to kill the virus. And the other question that companies, especially food companies or any company that still have workers operating and manufacturing whatever they are manufacturing, is what happens if what you need to do if you have a sick worker. And there's different recommendations for how much time you would consider that another worker would be exposed to the sick worker. And there are different recommendations that sometimes they're not based on evidence. Because, for example, okay, you are in close contact to a colleague that is working nearby, less than a meter from you, or about a meter from you. And if he eventually got sick, was I exposed by being in proximity to him? So that's some of the questions. There's still we're trying to struggle, and some of the earlier research is helping, but we still in many ways a lot of it is you don't really know whether at what point, for example, whether by speaking we're going to be expelling the virus to the air, and we're going to be causing an aerosol. Who knows? Anyway, do you want me to go on? There's this only slide. This is the only other slide that I have for.
So, Doc, we've got some questions here in the chat box. You can read them, or I can read them to you. There are 3 questions already. And I think maybe we address that and then see what we still have time for.
Yeah, no, that's fine. I don't know, how much time do you want to, when do you want to end this session?
We've just got a few minutes left. So.
Oh, you have 40 minutes?
Yeah.
Yeah, it's one, right now I have almost 1:30 here. Okay. So let me, okay, why don't you read the questions if you are seeing the questions, because I have my...
Okay, so the first question is from Ali, and he says, what measures should we adopt when we bring vegetables home for cooking in this current situation?
What measures? That's one of the points that I was going to touch next. So it's it depends on the source of the vegetable first, but if you're going to use your vegetables to cook, probably you should not have any concerns. The issue would be much more if you're planning to eat them raw, because but if you're going to cook them, then that's not going to be an issue. The only thing that we're recommending for people is to wash your hands as often as possible, and make sure that you sanitize them when you're in contact with other surfaces that may have been touched by someone else. But cooking is your best friend. Now, if you're dealing with fresh vegetables, what we're recommending is just wash your vegetables with water, I guess, for the most part. And this is an area that we don't have a lot of data what do you do with your vegetables? Well, I guess right now, probably just plain water should be sufficient, but we don't have data, unfortunately. This is the part that we are talking with the media, or and then they want us to recommend consumer what to do. And some consumers are taking they're taking the problem in their own hands, and some of them are actually washing with soap. And I think it's not something we recommend normally for food safety purposes. But all right, I'm back.
Hey, Doc, thank you so much. I'm sorry. I think there's a Zoom usually allows 40 minutes, and this looks like it's canceled on the 30th minute.
Oh, okay. That was very quick.
Sorry about that.
No, that's not a problem. I'm glad that we're back. Yeah, I think we were talking about the question on vegetables. Did the questions disappear, unfortunately? Yeah, it looks like the questions disappear. But yeah, that's the vegetables. I was saying that some people are washing with soap, but we normally don't recommend you do it. But if you happen to do it because you want to feel more comfortable, just rinse it well. That's my only additional recommendation. Do you remember any other question?
I've just
Wild animals.
Yeah.
Well, yes, I think the question about the relationship of wild animal meat and zoonotic diseases, I think this is something has been established for many years. I guess humans continue to interact with and invade different natural habitats of multiple animals, and animals, we do carry viruses that could be could jump the species barrier, and eventually we have things like what we're having right now. And yeah, the more traditional zoonotic organisms, for our case, some of the zoonotic foodborne pathogens like Salmonella, E. coli 157, they reside in animals and also in wildlife, some of them. So it's something that I guess I hope that humans, we learn the lesson after this pandemic that that we should try to keep our if you're going to eat meat, that try to keep it to domestic animals and don't go out there. I know that in some cultures, so the wild animals is something that is a delicacy, but this is a really hard lesson that we should not be doing this. But anyway, that's my own opinion.
Okay, then we have a question from Ella. I've unmuted her mic, so let's see.
Okay.
There.
Hi, Dr. Diaz. I'm a student in Virginia, and I've been helping with our local COVID-19 testing efforts. And we've had a number of farmers come through who've been told that they aren't allowed to either sell their products or milk trucks won't come to dairy farms to pick up the milk. I was wondering what you'd recommend that we do to help our local food supply and our local farmers when they run into these issues.
Okay, those are much more complicated issues than what I can advise, because I'm a scientist with a relatively narrow field in food safety microbiology. What you're talking about is beyond what I can recommend. Yeah, the challenges that we're facing with the excess of food that have to be dumped because the distribution channels have been interrupted and the demand has declined. I'm not sure how to be able to address your answer your questions. It's really a major problem, and we hope that the disruption of supply chains is not going to continue, because then things can get really more complicated in terms of food security for multiple populations. Like what you're hearing and reading in the news, some of the closures by large meat plants and the implication that may have for the entire population. So it's, sorry, but I don't think I can help you in this question.
Okay, we've got another question that says, what about the processing of offals as a food item? What is doctor's stance on that?
Sorry, can you repeat the
what about the processing of offals, like internal organs and stuff like that, as a food item?
I know that different cultures like to eat different parts of the animal. I think I would say cooking for viruses, cooking is your best friend. It doesn't matter what part of the animal you're going to be eating. As long as you cook it and you don't eat it raw, probably going to be fine. But if you're talking about the kind of a concern like it used to be, transmissible spongiforms, then that becomes an issue. What part of the animal you can or you should not eat. So, but that's a different organism to the one we're dealing with. If we just restrict our scope today to talk about viruses or coronavirus, I think it's yeah, with any animal product, as long as you pasteurize it or cook it well, probably should not be a problem.
Okay, and then we've got one last question before Andreas chats to us again.
Okay.
The question is, if we bring lemons from the market at home for making its juice, as doctors recommend lemon because it contains high vitamin C, which boosts your immune system to fight against COVID-19, how do we process it?
Okay, well, with lemons it's easy because they have a peel or yeah, the outer shell. So because you can then wash the lemons, you're not going to eat the peel, so you can wash it, and the virus is going to, if hand washing works for getting rid of the virus and killing it, because the virus is quite susceptible to soap because of its lipid bilayer and its coating. I think, yeah, wash your lemons before you cut it, and then should be fine. Those are really the relatively easy case to deal with. Other
Okay, I think that's about it. I think I'm going to hand over to Andreas right now. But Doc, thank you so much for your time. This has been quite a rewarding experience, and I hope that your research gets to move a little forward, a little more faster for COVID. Ooh, I don't know if we can all hear you.
Thank you. Thank you, Dr. Gonzalez, for this presentation and helping us this starting with these webinars. And the World Food Safety Day is coming, and I would be happy to have you there and help us with this campaign too. And I invite you everybody to the next webinar we have next Saturday, the same time, which is with Dr. Cheryl, the Executive Director of One Health Commission, and it's about One Health and COVID-19. Thank you for participating.
Bye, everyone. Have a good rest of the weekend.
Thank you again. Thank you to all our participants as well.